Hello friends, today we are going to learn differential RNA processing topic. What is differential RNA processing? We have already discussed about C-value and C-value paradox. Initially the concept of C-value came into existence and the scientists think that as the complexity of an organism will go on increasing, the DNA content will also go on increasing. Correct? Because more the complexity, more is the characters. But very soon it came to know that the complete DNA, the complete genomic DNA present in an organism is not a coding one. There might exist much more non-coding DNA, that is junk DNA, other than the coding one. And later on they came to know that the amount of non-coding and non-functional DNA is actually much more than the amount of coding DNA, and a new concept, C-value paradox, came into existence. But this thing is not going to stop here. There are much more things beyond this. Okay, so apparent complexity does not correlate with the number of protein coding genes. Look at here, that the apparent complexity, okay, it does not correlate with the number of protein coding genes. We are not here talking about the junk DNA, but we are talking about the protein coding genes even. Okay, means a single gene can give rise to various products. If this is so, definitely if a single gene can give rise to many products with every changing environment, with every changing condition, the same gene will give rise to various products with the changing environment or with the changing conditions. Correct? And that's what we are going to learn today. So, even the amount of genomic DNA, okay, that is also not going to correlate with the apparent complexity of an organism. Okay, and mainly two important concepts came into existence in this. First is a alternate splicing, and second is a poly A site choice. Okay, so let's move to the alternate splicing concept. Up till now, what we have discussed, that we are well familiar, most of the genes in eukaryotes are split genes. Split genes means you have, other than exon, you also have introns. Okay, and there might be many introns. Of course, the length of introns and the quantity of introns is much more generally than exon, and we have discussed that these introns are released, they are removed by splicing, and only the exons will join to form functional mRNA. Correct? But suppose the way these introns are removed and the way the exons are joined, okay, will also have different mechanism. Definitely it will give rise to various products. Example, if this exon 1, exon 2 and exon 3, these three exons appear, and these are the two introns, intron, correct? Suppose E1 joins to E2 and then E3, definitely it will give rise to a different transcript, E1, E2 and E3. It will be a different mRNA, functional mRNA. But what if the E1 will directly go to E3? This transcript will be smaller as compared to this one, but definitely the product spot will vary. Means the same gene can also give rise to different proteins, and that's what we are going to see, and this is referred as alternative splicing. Okay, so in yeast, the number of genes showing alternative splicing are less, but in human genome, more than 95% of the human genes show alternative splicing. Means actually the number of genes are less, but the number of proteins are much more than that. So primary transcript itself contains some molecular signals, okay, for alternative splicing. Means this primary transcript, that is pre-mRNA, non-functional mRNA, which has introns along other than exons, okay, it itself will be having some molecular signals for such alternative splicing, okay, and pathway favored in a given cell or metabolic situations, okay. So which type of condition, okay, a cell will get, according to that which type of splicing pathway, okay, or which type of protein will be formed, okay, that in a given cell or metabolic situation, okay, is determined by processing factors, okay. So the processing factor means spliceosomes. For example, protein composition of snRNPs, yes, small nucleotide ribonucleoproteins. These snRNPs, which are involved in splicing, which are a part of spliceosome, their composition varies with the changing condition, okay. So snRNPs in spliceosomes may vary, so that processing of a particular gene is altered in different stages of development, and it has been observed that different stages of development will show different composition of spliceosomes, which will identify different splicing sites, okay. Three different forms of myosin heavy chain, three different forms are formed at different stages of development in a fruit fly, okay. So in a fruit fly, during the different stages of development, it will form three different types of myosin, but the gene is same, okay. So let's move towards what exactly going to happen in alternative splicing. Here we have taken an example of alternative splicing, and look at it. Here we have taken an example of two different 3' splice sites. What is this? Look at it. This is your DNA, and of course this is the 5' and this one is the 3'. Okay. This is the 5' splice site. Okay. So towards the 5' end, this is the 5' splice site, correct? And this is the 3' splice site. But in this gene, two 3' splice sites exists. Okay. One at this site, second at this site. Result of this is because of these two, okay, splice sites. Splicing can take place either here or here. Okay. So after that there is one more part of the DNA, and this is the poly A site. Okay. At this stage you have poly A site. Correct. So now what will happen? Of course, after transcription, capping will take place. Correct. So this is your primary transcript, because still it is not having polyadenylation or splicing. Okay. So after cleavage and polyadenylation, sometimes it will show cleavage of only this much part. Correct. If only this part of the intron, if it is removed, definitely it will give rise to such a mature mRNA. Okay. With this region and this region. Correct. And as this is the poly A site, clear, this is removed and polyadenylation will take place. Similarly, if instead of selecting only this portion, if it will select this much, because the 3' splice sites are different. Okay. So if this is removed, a big part is removed and a comparatively smaller mature mRNA is formed. Cap is there, so translation will take place, and after translation, definitely the proteins formed in both the cases will be different. Okay. Here, through diagrammatic representation, I have shown the various types of splicings, okay, which can take place, okay, of the same gene. Correct. Look at it. Very first thing is alternative exon, which we have already discussed. Look at it. Consider these are the exons, and this thin region which we have shown is the intron one. We want to remove intron, and of course we want to join exons to form a functional mRNA. So these are representing the splicing area. Okay, actually at what site the splicing is going to take place, that is being represented by these, okay, regions. Now you can see, suppose this part is removed, and after that this exon, okay, is also removed along with, because splicing will take place at these two regions. Okay, so if splicing will take place in these two regions, definitely this exon is also removed, and this 1, 2, 3, 4, these 4 parts will join. Look at it, 1, 2, 3, 4, these 4 parts. Okay, but in the second type of splicing, of course, in a different conditions, when the conditions will vary, at that time this alternative splicing will take place. So at that time only these introns are removed and all the 5 exons are joined. So here you have a smaller functional mRNA, here you have a comparatively longer functional mRNA, correct? And this is the cap which is shown by red. Okay, this part is a capping region. Okay, and this cross is representing the polyadenylation site. Okay, this green cross is representing the polyadenylation, and that's why polyadenylation will take place in both of them, as this region is present in both of them. This is because of the alternative exon. Either the exon will be present or absent. Correct? Now, second is the alternative 5' splice site. Okay, so as usual, this is the 5' and this is the 3'. Correct? Again, you have 1, 2, 3, 4, 5, 6 exons, and you can see here that towards the 5' site, okay, either this exon will be removed or it will be included. Correct? You can see only the splicing can remove only introns, or sometimes this, because this is also a site for splicing. Correct? One extra 5' splice site is there, and that's why you can see if these all regions will join, definitely it will give rise to a comparatively longer mRNA. But if splicing will take place at this and this region, definitely this exon, which is shown by shaded region, okay, will also be removed, and result of this, if it is removed, definitely it will give rise to a comparatively smaller mRNA. This is alternative 5' splice site. Correct? Then alternative 3' splice site. Actually, we have discussed that here. Okay, you can see here that again you have this exon. Okay, and here there is a, towards the 3' end there is a splice site in between this exon. Okay, and result of this is, if it is spliced out at this region, okay, here and here, definitely this much part is removed. Correct? And only these 1, 2, 3, 4, 5 parts will join to form a mRNA. Similarly, if splicing will take place at this region and this region, definitely this intron will be removed only, but this complete exon will be included, and a comparatively longer mRNA is formed. This is alternative 3' splice site. Then look at it, it is an intron retention. Means it is not necessary that always the full introns are removed. Sometimes the intron is retained. Look at it. Here we have shown that this intron is retained, okay? Rest of the introns are removed, but this intron is retained here. What is intron? Of course, it's a sequence of ATGC. In case of mRNA, AUGC. Correct? So here, from the primary transcript, though other introns are removed, but this intron will exist, giving rise to, definitely it will be read by your ribosomes during translation, and that will also give rise to a comparatively bigger product. Correct? Similarly, if all the introns are removed, definitely it will lead to smaller mRNA and comparatively smaller protein. So intron retention is also there. What is the motto? Okay, to form a comparatively bigger protein, is it the only one? No, definitely some coding regions must be there in intron also. Must be having some different function. Correct? So, is it that all introns are not non-functional? Some introns which are written sometimes in certain special conditions, in certain specific conditions, correct? They might be coding also. Correct? Giving rise to comparatively bigger mRNA. Then, mutually exclusive alternative exons. Look at it. What is mutually exclusive? Means there are some exons which are specially, mutually designed for alternative exons. Okay? Means they will be present in one product and they will be replaced with some other exon in some other product. Look at it. This 1 and 2. We have shown here number 1 and number 2. These two, okay, exons, they are alternate with each other. So, if this number 1 is present, number 2 will be spliced out. If number 2 is present, number 1 will be spliced out. So, it's alternate. That's it. Correct? Giving rise to two different mRNAs. Then, again, you have alternative promoter and first exon. Now, this is a very important thing. Look at it. What is going to happen here? This is your first exon. Okay? But this exon will be used only in one mRNA. This much portion is removed. Okay? If I will splice out this much portion. Definitely after this exon will join. Correct? This, and then this one, and poly site is there. But sometimes instead of using this exon, directly the second exon will exist. So second, okay, this is the third one, and this is spliced out for the new one. So definitely in this case, you have the first exon is alternative. Okay, but it has been observed in such exons in which the first exon is alternative, because this is the first exon in this mRNA and this is the first exon in this mRNA. In such case, these exons also have some promoter regions in them. Okay? So, either I can say alternative promoter and first exon. So, promoter region also exists in them. Correct? And this is how the capping and the first exon will vary along with promoter, and poly A tail is as it is. Correct? But other than that, one more important type of splicing which will lead to a much bigger variation in the two mRNA is alternative poly A site and terminal exon. Okay? So here the poly A site is alternative. Correct? In that, this is your the pre-mRNA, primary transcript, and you can see here and here I have shown two crosses. This first cross will represent the first poly A site. Okay? So definitely this much part can be spliced out. Okay? This much part will be removed, and definitely you will have 1, 2 and 3. These three regions join, and here there is a poly A site, so polyadenylation will take place. But alternatively, you have here also poly A site is present. In this case, this region is spliced out. So this shaded exon will not appear, and these 1, 2, 3, 4 will join. Correct? And poly A site as it is present here also. Clear? This will be. So here in these two cases, okay, especially in these three cases actually, in which mutually exclusive alternative exon, alternative promoter and first exon, and alternative poly A site and terminal exon, in these three clear cases, you will find that the mRNA, the functional mRNA which is formed, show much more variation than rest of the alternative splicings. Now, another important processing of RNA transcript which leads to differential RNA processing and leads to different types of mRNA formation is poly A site choice. We are well familiar. One of the important post-transcriptional change is 3' polyadenylation. Continuous adenine residues are added, correct? On or AMP are added on the 3' site, correct? This is for protection of mRNA in eukaryotes. But the poly A site, that signal which is involved in location of the site for polyadenylation, is also present more than one times. So definitely, if in a particular mRNA, if more than one times the poly A signal is present, then cleavage and polyadenylation can take place only on this much transcript, or cleavage and polyadenylation can take place on this much transcript also. Means again it will lead to formation of different types of transcripts, correct? So first of all, the poly A site choice, okay, takes place in complex transcripts, okay, only complex transcripts, and 2 or more sites of cleavage and polyadenylation will lead to what? Different types of mRNA. Again, different functional mRNA means different types of proteins. Then, for example, the best known example of it is variability in domains of immunoglobulin heavy chain. So we have learnt in immunology that various types of immunoglobulins are there. IgG, IgA, and in that only a single gene is present for the heavy chain. But the single gene leads to various types of antibodies and a big diversity amongst that. Correct? And this all is because of the polyadenylation site, other than the various splicings which are also involved in that. But polyadenylation site, the different polyadenylation site are having prime importance in that. Okay, now look at this picture. This is your DNA, and you can see this one is your exon, correct? And 2 poly A sites are present, one at this site A1 and this is another one A2. So there are 2 poly A sites, and after that again there is this exon, correct? Now after transcription you will form the pre-mRNA. So this structure is your pre-mRNA, and on the pre-mRNA, clear, if the cleavage and polyadenylation takes place on this site, definitely only this much transcript will be formed, and again this is the polyadenylation site, and that's why polyadenylation will take place. But if it will take place at this site, definitely it will lead to formation of this part of the mRNA. These the 2 different mature mRNA are formed, okay, and this is how clear the poly A site choice is there. Of course, as this is a long mRNA, definitely it will lead to a big protein formation, and this will lead to a small protein formation. So in this way, because of poly A choice also, you can form different types of RNA molecules, because these different splicing and different poly A sites, they lead to formation of various types of mRNA. So gene is the same, but products are different, correct? So one of the very common example of this, okay, which lead to different formation of different types of proteins, is about both alternative splicing and poly A site choice process a single mRNA transcript to produce 2 different hormones, and the best known example is of calcitonin. Okay, calcitonin, which is a calcium ion regulator in rat thyroid. Okay, but same gene required for, or which is responsible for formation of calcitonin, also lead to formation of CGRP. Okay, what is CGRP? Calcitonin gene related peptide, CGRP, and this is formed in rat brain. So same gene lead to formation of calcitonin in thyroid, and the same gene CGRP, okay, same gene will lead to formation of CGRP also, but that in rat brain. Clear? And CGRP is a hormone as well as a neurotransmitter. It is also involving the thermal homeostasis. Okay? Then, other than that, it also regulates the heartbeats. Okay? And so many other things. Then, this is actually the example, clear, of calcitonin, which we have discussed just now, how a single gene can lead to formation of different products. Okay? Now, in this, here we have shown the primary transcript. Okay? And you can see, this is a cap which is 4, correct? 5' cap. So, this is the 5' end, no doubt. And of course, this will be the 3' one. And in that, you can see this is the first exon, then intron, second exon, intron, third exon, intron, and this fourth exon, this is actually coding calcitonin. But here is one polyacide, correct? Another intron, and this exon number 5 lead to CGRP. Okay. After that, what do we have is again exon 6 and another polyacide. So you have two polyacides, one after the sequence 4, exon 4, and second after the exon 6. Okay? And this is the primary transcript, and cleavage and polyadenylation in case of thyroid. Okay? It will take place at this site, and because of that, only this much mRNA will be formed and polyadenylation will take place. Correct? Of course, later the intron parts are removed by spliceosomes and only the exon 1, exon 2, exon 3 and exon 4 is responsible for calcitonin, correct, are joined, correct, forming mature mRNA. After translation, it will lead to formation of this much, clear, the polypeptide. Later, after translation, the post-translational changes will take place. During that, the protease will cut this site, releasing only calcitonin. On the contrary, this is in case of thyroid, but on the contrary, in case of brain, the process is quite different. Because here, this is the polyadenylation site. Result of this is we form this much amount of mRNA and polyadenylation will take place, correct? But after that, don't forget, these are the introns, okay? Intron, intron, intron and intron. So definitely, no doubt, cut will take place at this region. And after that, clear, here, at this as well as this, both these regions the cut will take place, and result of this complete part is removed. Okay? This complete part means this exon 4 required for calcitonin formation is also removed along with these introns and these introns. Correct? So what will join is E1, E2, E3 and directly the sequence 5 and E3. This is your mature mRNA, and after translation it will lead to formation of the polypeptide, and this polypeptide with the help of protease action will be cleaved to release only this CGRP. So in this way, CGRP is formed in brain while the calcitonin is formed in the thyroid of rat. But the gene responsible is same. So initially, long ago there was a concept, one gene, one function. It failed because to carry out a single function in our body, we require more than one genes. So one gene, one function? No, not possible. Then a new concept came into existence, one gene, one protein. But later on we came to know, for a single protein, we have more than one polypeptide, and these polypeptides are coded by different genes. And that's why that concept also failed. New concept came into existence, one gene, one polypeptide. But now, from a single gene, you generate different polypeptides also. So, is it possible that one gene, many polypeptides? can also be the concept. Okay, so many things are there, clear, in molecular biology, clear, that we can't predict only a single thing. Okay, we have many things to study in mol bio. So what we have learned today is the differential RNA processing, and in that we have gone through the various types of alternate splicing. We have seen so many mechanisms, and the poly A site choice. Because of these two important processes, from a single gene, various proteins or polypeptides can be formed. Thank you.